Hi everyone, welcome to my channel Srinivasa Biology Tutorials. For this session to discuss NCERT Class 12 Chapter 4 Principles of Inheritance and Variations Part 2. As Mendel found the phenotype of the F1 heterozygote, that is the capital T and small t, to be the exactly like as in a capital T small the capital T parent to get appearance, he proposed that as in a pair of in a dissimilar factors. So when you observe as in two dissimilar factors, how do they are taking here? Then in a parental characters, these are in capital T and small t. These factors, these are completely to get producing as what in a heterozygous characters. Here one is the dominant, another one is in that recessive, that is a F1 generation, it is in a dominant, another one is in what, that is that in a recessive parent. See, in this case of the capital T is a dominant over the small t is a dwarfness, it is called as a recessive parent. So just we see that one only, it is in a capital T and it is in a small t. He observed as in the identical the behavior for all the other characters, the trait space of their study. So he studied about as in the characters, how do they complete the cross with as in the dominant and recessive characters. He observed. Okay, and let us. It is the convenient to use as in the capital and lower case of any the alphabetical symbols. The remember is in a concept dominant and the recessiveness. So here when you take as a one capital T, small t, it is that show as the dominant phase, it is show as in the recessive phase. So it is easily to completely to get determined as in a that characters also. If in a case don't use as in a capital T and small t for dwarf cases, you will be there find that difficult to the remember what their capital T's and small t allele in the same gene characters do not. So when you observe here capital T and small t, here like don't use, because when you do like what happened it make as in a confusion. So that purpose don't use that one. Here allele can be there similar in the case of the homozygous capital T and then small t can be the dissimilar as in the case of other heterozygous character, that is in a capital T and then small t. Since here the capital T small t plants, here the capital T small t plants that heterozygous character for as in a gene the controlling one character, that is in a monohybrid cross, and the cross between the capital T small t that is in a monohybrid cross. So we are getting as in that capital T the dominant characters small t that is in a the recessive parents, we getting as in that what it is, we get as in a the first the F1 generation the heterozygous characters we are getting. From the observation of the recessive parents, the traits are expressed without any the blending in the F2 generation. So when you observe here the capital T small t, in this capital T small t, we are getting as in that gametes. Am right? Here when you cross here capital T small t, these gametes are not blending in the F1 generation, the F2 generation. So when you refer to as in F2 generation, what happened? Again the characters will be there expressive. Here like as in capital T small t, capital T small t, and then small t, like the characters will be the get re-expressed. Okay, and then here we can there infer that when the tall dwarf plants they produce the gametes by the in a process of the meiosis, the alleles, the parental pairs separate and segregate from as each other of one by one. The allele is the transmitted the gamete. So in that meiosis process once it takes place, what happens? The gametes separation will be also there takes place. The segregation of the alleles, the random process, and so they are in a 50% of the chance their gametes they containing either allele has been verified that in a result in a crossing. So when you crossing here, what happened? The mostly the 50%. See here in the F1 generation, the 50% of the characters comes from as a parent. So capital T 50% and then small t 50%. In this, why the in a gametes of the capital T, the plants have in a allele. The capital T is gametes of the dwarf in a small t plants also have in that in a the allele that is in a small t. So one is a completely capital T, one is in a what? Small t. Next, after during the fertilization, the two alleles capital T's one parent set through as in a pollen and the small t another parent through as in a egg cell that united the produced the zygotes have one tall and one more is in a dwarf. So here when we are taking as two gametes, when the two gametes we are cross, what happen? We getting as what? In a zygote. Here for example it is in a male gamete, it is in a female gamete. Here we getting as what? The zygote will be there coming here. And another word hybrids there is a capital T small t. So when you observe as in a capital T small t line. And since these the hybrids contain alleles which express as in a the contracting traits, that is a heterozygous character. So here one is a capital T small t is there. So that is character is called as what? That is in a heterozygous characters. The production of the gametes by the parents, the formation of the zygotes, the F1 and F2 generation plants can be they are understood from the diagrammatic representation, the Punnett square. It shows as in a figure 4.4. So when you observe here the Punnett square also they given. So in the right column and the left column to given as in the heterozygous parents means capital T small t, capital T small t. When you get draw here, we get cross here. What happened? Capital T, capital T. We are getting as in a capital T, capital T. It is in a capital T small t, and here getting as a capital T small t, and we are getting as in a small t, small t. Okay, it was developed by as in a British genetics that Reginald C. Punnett. It is the graphical representation to the calculate the any probability all possible the genotypic offsprings in the genetical cross. So how do the genes they are getting in the generation to generation? He clearly explained about as in a one graphical form. That is in a nowadays we are calling as in what? That is a Punnett square. Okay, and the possible of the gametes are in a written in a two sides of usually top row and left columns as in a possible the combinations are represented in the box below in the squares, which is generates the square output form. So when you observe as an output form, we getting as this all characters. Okay, let us. The Punnett square show that in a the parental characters capital T male and dwarf small t the female plants. The gametes produced by Zinni that them one is a capital T small t in the F1 generation progeny. The F1 plant, the genotype capital T small t as in self-pollinated. That symbols is a female and males are used donated as in a the female egg cells and male that is in a pollen grains. In the F1 generation, in the separatively we getting. So in that capital T small t we get as in a F1 generation. Okay, now after in that F1 generation of the understood is a typical monohybrid cross, the self-pollinated. It is in a completely the capital T, small t gametes only they produced as in what? Gametes. In that capital T small t, it is in a they introduced by as in that Mendel the proportion. So Mendel proposed these characters. When the fertilization is takes place, the pollen grains they are germinates to get produced as a true breeding tall plants. Capital T is in a 50%. And the pollinated eggs, their genotype and as well as to get forms as in the genotype also it is in a 50%. So 50% in a capital that is a dominant characters and 50% of in a completely what the pollen grains, the small t also they get produced as in the chance will be present. Okay, then the pollination of the eggs of the genotype. See of the pollinating eggs the genotype capital T as well as in a pollen type that form of as well as in a genotype the small t as a result the random fertilization it is forms as a zygote to get genotype what is that capital T small t and then small t will get coming. Okay, this is and after when you observe here the fertilization leads to the capital T of leads to as in a capital T small t 1 by 4,000 and small t through in the F1 have the genotype of capital T small t but the phenotypic character seen that in a tall in F2 generation 3 by 4 of the plants are tall. So when you observe to as in a 1 by 2 and they are completely 1 by 4 and 3 by 4 characters we are getting in that Punnett square. We are sum them in capital T and otherwise in a capital T small t. The exactly it is not possible to the difference between as in a plants while that in a genotype of capital T or small t. So when you are how do we get identified which one is a capital T, which one is a small t? It is not possible in that capital T, and then capital T, small t. Hence, within the genotypic, the piece capital T, small t, in one character, the capital T is tall expressed. So, hence, these characters or capital T is tall said to as a dominant over than the in a allele, that is in a small t or dwarf character. So, it is in a dominant character, it is in a what? That is in a recessive parent. This is thus the due to the formation of the dominant one character over other than the F1 all through the genotypes capital T, small t in the F2 generation 3/4 in plants, the tall 1/2 as in capital T, small t, 1/4 is in a capital T, their forms. The leads the phenotypic ratio is in a 3/4 in a tall, 1/4 in a capital T, and 1/2 is in a capital T, small t, and 1/4 is in a small t. So when you observe to as in a ratio, here the phenotypic ratio is, that is in a 3:1. The genotypic ratio it is in a 1:2:1, getting as a 1:2:1. Okay, let us, after here the formation of the 1/4, the 1/2 and 1/4 of in capital T, one capital T, small t, the mathematical considerable the form of in a, the binomial expression, that is in a ax plus by square, that is in a gametes, the bearing as in a capital T or small t, the equal frequency of 1/2 expression of expressed given below. The reaction is 1/2 capital T, 1/2 small t whole square is equal to 1/2 capital T plus 1/2 small t into 1/2 capital T plus 1/2 small t is equal to 1/4 capital T plus 1/2 capital T small t plus 1/4 small t. It is in a completely mathematical formula. Okay, remaining to discuss in next class. Thank you for watching.